In this set of notes, we're going to be looking at an introduction to what's called genetics, or how traits are passed from parent to offspring. Today we know that DNA exists, and we know what it does, which we've talked about in previous lessons. But the ideas of genetics and heredity came long before we even discovered DNA. Genetics is defined as a branch of biology that studies heredity, and heredity is the passing of traits from parent to offspring. This man here in the picture, his name is Gregor Mendel. He's known as the father of genetics. He was born in Austria, and he became a monk. A monk is a person who lives in a monastery. They spend their life worshiping God, and they live a very poor type life, but they do a lot of good things for their community and things like that. Well, anyway, his parents had an agricultural background, and he was very interested in plants. And a botanist is someone who studies plants, so we could even say that he was a botanist. He's called the father of genetics because he spent so much time working with plants, and he discovered the patterns of inheritance that we know today. Specifically, he worked with the organism here in the picture. This is a pea plant. He grew peas in the monastery gardens. I suppose they probably ate them, And he kept detailed records of the pea plants across many, many, many generations, because he lived in the monastery, most of his life. Plus, you can grow pea plants pretty quickly. Using statistics, he was able to predict the outcome of different pea plant crosses, where you breed the different pea plants. And he established new and exciting theories about inheritance patterns. So why pea plants? Well, for one thing, the male and female parts of the pea plant are on the same flower. So it can be very easy to control mating. If you don't want the flower to self-pollinate, for the pollen to fall on from the male part of the flower, the stamen, onto the female part of the flower, which is called the pistil, you can just cut it off. Okay, so you can cut off the little stamens that make the pollen with some scissors, and then you can take them over to another flower and pollinate that flower with the stamen that you just cut off. So you can control which plant mates with which other plant. Peas are also very easy to grow. You can grow them, in a little pot by your kitchen sink. They don't take very much room to grow. They are very hardy little plants, and they also grow very fast, and they produce lots of offspring. As you can see in the pea pod there in the picture, each one of those seeds is considered one offspring. So just one pea pod, it's got like 8 offspring there. So just one pea pod would have 8 offspring. Pea plants also exhibit characteristics that come in 2 different forms. So here in the picture, you can have either a white flower or a purple flower. Other characteristics that Mendel looked at for the seeds, they can be different colors. They can be yellow or green. The seed can also be nice and round, or it can be wrinkly looking. Of course, we just said flowers. They can be white or purple. The pod can also be yellow or green, and also full or kind of constricted or tight around the pea pod, around the peas themselves. And then the stem, they can have long stems or short stems. And the placement of the flowers, they can either be like at the top of the stem, which we call terminal, or they can be down inside the stem further, which we call axial. Most of what we're going to be looking at is either the flower color, the seed color, or whether the seeds are round or wrinkled. Okay, so here's what Mendel would do. So let me get my little mouse on here. So here we have the flower, okay? And it has both male and female parts on it. And you can take the scissors and cut off the stamen. The stamen is the little yellow part that makes the pollen. Then you can take that pollen over to another flower, or you could do it the other way, okay? And you can even do that with a paintbrush. This has a paintbrush here. You get some of the pollen on the paintbrush and you take it over to the other flower. So this is how he can mate them. He can control which flowers are mating with which flowers. Okay. Now this type of cross where you mate where you have one characteristic where you're just looking at like flower color is called a monohybrid cross. We're just looking at one trait, just flower color. We're not looking at seed color. We're not looking at seed shape or height or anything like that. Just one trait is called a monohybrid cross. And that's going to be important. Okay, so this is maybe a better way to look at the inside parts of a flower. This is the girl part here called the stigma. And these two parts on the side are the boy part called the anther. And this is what makes the pollen. Okay, so over here, the boy part here is the lower section. They're both brown kind of, but like A, and the B part as well. That is your boy anther, and it's held up by the filament. Then your girl part here is up at the top, the C and D part. This is called the stigma, and the little part that holds the stigma up is called the style. So again, if you want to control, this is a lily flower, you want to control the mating of this lily flower, all you got to do is cut off the anthers. I know that kind of sounds like mean, right? Like you're cutting off part of the flower. But they actually do that even for weddings because this makes a lot of pollen, and brides and flower girls, they don't want that pollen on their clothes. And so a lot of florists, if they're using lilies, they'll actually cut those off if they're using them for a wedding because they'll get yellow or brown pollen everywhere. Okay? But anyway, that's another way. That's just a way that you can control how it mates, because if we leave it on there, then this pollen can just go right up here, blown by the wind or by a bee or an animal might carry it up there, and the flower will just mate with itself and you'll get an identical flower because it used its own pollen, right? But if we want to mate it with, say, a white lily flower, then we need to cut those off so that we're ensuring the pollen that it's getting is the one that we want. Okay. So that's what he did. He used, sometimes he would allow the plants to self-fertilize, or to just let the pollen go from the anther up to the stigma. But other times he would cut the anthers off. Now, one important thing is the difference between a true breeding plant and a plant that is not true breeding. A true breeding plant, so say for example it has purple flowers, a true breeding plant will always make purple flowers. If you let it self-pollinate, you don't cut the anthers off, you don't dust it with other pollen, and it only gets its own pollen every single time, it will always make purple flowers, and that's what we call true breeding. However, if you have a plant and it looks purple and you let it self-pollinate, and the offspring, the new plants that grow, there's some purple and white flowers. Well, that is not true breeding, okay? So a lot of times when he was doing his experiments, he would start out with the true breeding plants. And those would become what we call the P generation, or the parents, okay? So here's a picture of that. So he's going to start out with a true breeding purple plant. It can only make purple flowers. And he knows that because he's watched it over several generations, and it only ever makes purple flowers. He's going to cross it with a white true breeding plant. It only makes white flowers. And these are the P generation, P for parents. He's going to let these two breed with each other. Now, the first generation, when he planted these, F means filial, which stands for son or daughter. That's what filial means. They were all purple. Okay? Then he let those self-fertilize. So he just lets them have their own pollen. He doesn't transfer pollen to them. And when they have babies, this is the F2 generation. This would be like the grandchildren of these. Okay, so these are the children, these are the grandchildren. Well, now we see a white one reappear. And it came up, when you do statistics, when you count them, you get 3 purples to 1 white. And he did this over and over and over, hundreds of times, sometimes even thousands. He would have thousands of pieces of data, and he would always get about this same ratio: 3 purples to 1 white. Upon reviewing the other traits, he also got 3 to 1. So if you look here, look, these are his actual results, okay? So 3.15 to 1, very close to 3. You can see how close to 3 all of these numbers are. They all round to 3. And these are all his data points, how many data points he had. Look at that. Look at the seeds. 6,000 seeds. He counted those things. Can you even imagine? But he always got this 3 to 1 ratio. Okay. So how do we explain this? How do we explain this white flower reappearing? How do we explain this 3 to 1 ratio? Because it looks like it's gone here, but then it reappeared. So what he said was organisms carry both forms, but one of these forms is like stronger and masks the other one. And we call that form the dominant trait. So here, purple is dominant over white. Dominant. We call these ones that are not dominant recessive. Dominant, recessive. These mask or hide this one in the F1 generation. Okay, so when we have these different forms of a gene, you know, we have the purple form or the white form, we call those forms of a gene alleles. And we get one allele from the egg, from our mom, and we get one allele from the sperm. And so again, those are called gametes. Egg and sperm are called gametes, and when they combine, when they combine through fertilization, they create a baby. And so again, you get one allele from mom and one allele from dad, the form of a gene. Now, as we get into this unit, we will be working problems with these pea plants and with other organisms. And so we come up with ways to represent these different genes. Now, a dominant gene, like the purple flower, is always represented with a capital letter. A dominant capital letter. So here, this is talking about tall versus short plants. So we're going to say big T equals tall. Now, we always use the same letter. We're not going to say S is for short. We're just going to make short the lowercase t. It will not be T and S. It usually always takes the letter of the dominant, and then the other one gets the lowercase letter. Okay, so and then there's 3 combinations. So you could get a big T from mom and a big T from dad. And so you would have 2 big T's. And so this plant would be tall. There's no other thing for it to be. It's just got tall genes. This plant here has 2 little T's. Well, there's not a big T there. It's got to be short. And this is called your recessive phenotype here. Here we've got one of each. We've got a big T and a little t. This plant will also be tall because the big T covers up or masks the little t. It's like it's hidden. It's like this one's stronger than this one and it covers it up. So this one is still tall. Okay, so we have 2 dominants here, 2 recessives here, a dominant and a recessive gene here. We got lots of words. Genetics has lots and lots of definitions, okay? So here's a list of some of those definitions. Genotype, that's your actual genes. That's the big T and the little t. It's what is in your actual DNA. That is the opposite of, if we come down here, phenotype is what it actually looks like. Excuse me. Is the plant tall or is it short? Okay, so again, genotype is the letters, the genes. Phenotype, physical and phenotype both start with F. The F sound, PH and PH. So they go together. Phenotype for physical. What does it actually look like? Is it tall or short? Yes, you need to know the difference between genotype and phenotype. Two other words that you need to know the difference of that kids always get confused: homozygous and heterozygous. Homozygous means you have two of the same gene. You can either have two big T's or two little t's. Homo, that root means same. Okay? Homo means the same. Same, same. If you have two big T's and they're both dominant, we call that homozygous dominant because it only contains dominant genes and they're both the same. Homo, same, dominant, two dominants. If they're both recessive, if you have two little t's, again, they're the same, so we still have homozygous. Now they're both recessive, so homozygous recessive. And then the last definition that we need to know is heterozygous. Heterozygous means you get one of each. They are different. The root of hetero means different. So here we have a big T and a little t. They are not the same. You have one gene for tall and one gene for short. However, this individual will be tall because, again, the tall masks or covers up the little t for short. Okay, so here's an example. Homozygous dominant, two big T's. The only possibility is to be tall. Homozygous recessive, again, homo means the same. Recessive means the lowercase letters, so they must be the same. There's no possibility of being tall here because there's no big T, so it must be short. Heterozygous, again, hetero means different, so we have one of each, a big T and a little t. There is a big T. As long as there's one big T, it's going to be tall because, again, that big T covers up or masks or makes the other one not look like it's there, however you want to think about that in your mind. The big T covers up the little t, and all we see is tall. Okay. It says determine each pea plant's genotype and flower color. All right. So we know that in Mendel's cross, he always started out with true breeding plants. So if this one is purple, I'm going to use P for purple. This one is going to be a big P and a big P. It has to be because it's true breeding. Okay. Again, we're not going to use W for white. We're still going to use P. Okay. We know again that it is true breeding. So now this is where it gets tricky. You got to make them look way different because otherwise, when you start doing the problems, P kind of looks the same, lowercase and uppercase. So you got to make them way smaller. Let's make this one even bigger. My line here. Okay, way bigger. Okay. Again, we know that he started out with true breeding plants. So the next one, well, you can only get one. You can get one gene from this plant. Say it's the mom plant. And one gene from this plant are going to make the baby. So the only possibility for this guy or girl is to be big P, little p. Again, I'm going to make them very different so that I can tell that it's heterozygous. Down here, I know that this one is white, so it has to be the recessive. So it's going to be two little p's. Now, your other three, we don't entirely know. You would have to, we know that they're all going to have a big P, right? Because they're purple. But there's actually another one of these, right? Because it's got to self-fertilize. So you got to have a mom and a dad plant, right? So what you got to do is, because it's self-fertilizing, or you could, anyway, there's got to be pollen, and there's got to be the egg that's inside the girl part of the plant, the stigma. So that at this level, there still has to be two. There still has to be a girl part and a boy part. So if we get a big P, say from this one, and it comes down here, But this parent, it could give a big P or a little p down here, okay? So if it gave a big P, we could get this. But if it gave a little p, we could get this, and it's still going to be purple. And the same thing with this one. We could either get a big P or a little p. Let's just say it's little. So we can get 2 little p's for this one, definitely. But these we don't entirely know. We can guess or estimate what they could be. But to know if it's 2 big P's or a big P and a little p, you'd have to actually look at the DNA, I guess. But in any case, you're going to have at least 1 big P in these because they're purple. Okay. How do I stop that? Okay, so this is called the Mendel's law of segregation, and this is one of the things that the theories that he came up with through all these experiments that he did. It's the idea that when parents pass on their genes, their gametes only get 1. So here we have the parent, its genotype and its genes is big T, little t. When it makes egg or sperm, depending on if it's a boy or girl, it can only pass on 1 gene to each gamete. And so this is called the law of segregation. Those genes, the alleles, separate from each other to make the gametes. Law of segregation. His other law is called law of independent assortment. What it says is that if you have 2 genes, so say you have flower color and plant height, they separate independently from each other. They are not like stuck together. So A can separate the big A and the little a, and B can separate big B and little b. But see, there's no like pattern to it. It's not like big A is always stuck with big B or little a stuck with little b, right? It's random. So this law of independent assortment is the idea that genes separate from each other independently. They're not stuck together. So now we got to look at another man. In order to work out genetics problems, we use a little diagram called the Punnett square. And this was developed by a man named Reginald Punnett. And you can see him there in the picture. And he was he attended the University of Cambridge and he earned a degree in zoology, and he later worked studying livestock. And farmers already knew that if they bred 2 animals, the offspring would receive characteristics from each parent. And he was able to determine patterns of heredity amongst these livestock, and he would chart out his experiments using what he called the Punnett, what we now call the Punnett square. And this is it here. It's just a box, and it is separated into 4 sections, okay? So here's an example of a Punnett square involving a plant. We're say this is our tall and short plant. We've got a plant with a genotype big T, little t, and a plant with 2 little t's. So here's that plant, the first one. Again, the genes are going to separate, so we're going to get a big T and a little t. Over here, we only have 2 little t's, so we write 1 little t and 1 little t. Now, to fill in the box, it's kind of like a multiplication table. So this big T right here goes in the box, and this little t goes in this box. Okay. Then over here, we have a little t. It's going to come in this box. And now I have to look over here and join them up. And that little t is going to go in that box. Next box, I have a big T. It's going to come down here. And this little t is going to come right here. Big T, little t. Then our last box, we've got a little t that can come down here and a little t that can come over here. A lot of times if I'm doing this on paper, I might use my fingers, and kind of trace where they're going to go so I know where the boxes are going to intersect, okay? or if you're doing it on the computer and you want to use a drawing tool like I just did, that can be helpful as well. I never know how to turn the little pin off. Okay, sorry. So in order to do a Punnett square, which we're going to practice a few in just a minute, there's some steps involved. First, you need to know what the genotypes of the parents are. Sometimes they're given and sometimes they're not. And if they're not, then you have to kind of figure it out. And I'll show you how to do that. Then you're going to draw your Punnett square. Then we're going to fill in the Punnett square, just like I just did. And then we will determine what are called ratios. And we are going to do that. Should you see one here now? Okay, it says the problem is up here in the grayish blue colored box. In pea plants, round peas are dominant over wrinkled peas. Wrinkled peas are recessive. Using these steps that were on the other slide, cross a plant that is heterozygous with a homozygous dominant. Okay, so here we've got, it says heterozygous. Hetero means different. I think that in my mind, different. So I'm going to write down two different, a big and a little. Then it's homozygous dominant. Homozygous means the same. Dominant means two big letters. Two big letters. Okay? Now I'm going to write those. They put the heterozygous here at the top, the big T and the little t. See big T, little t? The two big T's, you've got one here and one here. Then you're going to fill it in, just like before. So big T, big T. They go in that box. Little t, see the little t there? And the big T. And you write it in that box. And we nearly always write the, the capital letter first, if there is one. Okay, this box, we've got a big T coming from here and a big T coming from here. In our last box, we've got a big T coming from here and a little t coming from here. Okay, so we filled it all out. Now, determine the ratios. So let me see if I can erase all these lines because now I don't want them. Sorry, this makes it more confusing. So we want to know the ratios. First, we're going to find the genotype ratio. We're going to count the actual genotypes. So my genotypes that I see, I've got a big T and a big T, right? Big T, and there are two of those. 1, 2. There's two of those guys. These are also the same, big T, little t. Big T, little t. Also 2. So it's 50-50. Half of them are big T, and half of them are little t, little t. I mean big T, little t, excuse me. 50-50, and you can see that over here. That's what they got, 50-50. Okay, now it wants the phenotype. Phenotype's what it actually looks like. So I'm going to erase this again. So now we have to look at not the letters, but what do the letters stand for? So we know that big T, if there's any big T present, the baby will be tall. Okay? So if I look at this one, yep, it's a big T. That one's tall. Yep, there's a big T. That one's tall. Yep, tall. Big T, yep, tall. They're all tall, 100%. And there's the 100%. So again, genotype's looking at the actual letters. Phenotype is looking at what does it look like. Oh, I said tall, right, and they were doing seed color. I'm sorry, but it had T's. It should be for tall. All right, next one. Again, I'm not good at that. Okay. Okay, so here's one for us to work. In people, freckles are dominant. Use a Punnett square to determine allelic combinations among a woman who is heterozygous and a man who is homozygous recessive for freckles. Okay, so I'm going to use F for freckles. Says she's heterozygous, so hetero means different. So I'm going to have a big F and a little f. Okay. The man is homozygous recessive, so he has two little f's. Homo means the same. Recessive means little letters. Means, okay. It says freckles are dominant, so that means she has freckles and he does not. So now I'm going to write her up here. It doesn't really matter. This is my big F. Try and write them differently. Make my little f have a hook on it. Okay. And he is two little f's. Okay. And now we just fill it in. Okay. Big F. And then this little f comes over. And I'm gonna make it look more like a hook. Okay. Two little f's. We got a little f here and a little f here. Okay. I still don't like how this one looks. This is a big F. I think I'm gonna make my big F have like a darker thing so I can tell the difference better. Okay, so this one is the big F. I'm gonna make this darker because it just helps me see it better. And then we got a little f coming from over here with my hook on it. And again, two little f's in this box. Again, just do it just like you would a multiplication chart. You're just filling it in. All right, so now we want our ratios. So genotype, the only genotypes present are big F and little f, and two little f's, right? So it's 50-50. I've got two of these, two, and two of these. So my genotype ratio is 50-50, 50%. Or you could say 2 to 2, okay? It's 50-50 or 2 to 2. Sometimes they do it in percents. They'll say 50%. And other times they'll just count it and say, well, it's 2. You could put 2. And then they put this little dot, 2 to 2. Okay. Phenotype looks like it's also 50-50. Phenotype, let's do that one in a different color. This one has freckles. And so does this one. So both of these have freckles. Freckles. I'm not, okay. I'm terrible at this thing, you guys. Freckles. Yep, it's got the big F. As long as it has one big F, it's got freckles. Over here, no. There's no big F, no freckles. So it's still 2 to 2. It's still 50-50. Okay, so this one is also 50-50. No, they will not always be the same. And most of the time, they are not. Okay, so don't get it in the idea that it's always going to be that way, because it's not. All right, next. And I got to turn this thing off and then switch the slide. Okay. Albinism, or being an albino, which is where the animal is solid white, usually also has red eyes, is a disorder with a lack of pigment causing the deer to be white. Cross a homozygous dominant deer with an albino deer. Okay. So now, albino is recessive. So we're going to label it 2 little a's. Because it's recessive. If you're recessive, you've got to have 2 little ones, always. Okay, if you're showing the recessive. Then the next one is homozygous dominant. Homo means the same. Dominant means 2 big letters. So this is our brown deer. 2 big A's. So now I write that up here. Big A. Again, doesn't matter which one. Big A and big A. Little a, little a. And then you fill it in like a multiplication chart. Big A, little a, big A, little a. And look at this. What are you noticing? They're all the same, right? So this is gonna be 100%. I don't even have to count, right? 100% for both genotype and phenotype. You can also say 4 to 4. So again, it's gonna be 100% for big A, little a, and the deer color is gonna be brown because there is a big A. Now we call these guys carriers because they do have the gene for albinism, but they are not currently expressing it. But if they were to mate with another individual that was also a carrier, then they might have an offspring that has albinism. Albinism is common in all kinds of animals: deer, peacocks, squirrels, humans, rats, rabbits. Lots of different types of animals can have albinism. But again, this is 100%. You could also say 4 to 4. You can even say 1 to 1. They all would represent 100%. Again, 100% genotype is the A, big A, little a, and 100% phenotype is brown deer. Turn this pin off again. Okay. We got one more to go. Says fuzzy leaves are dominant over smooth leaves in sunflowers. Cross a sunflower that is true breeding for fuzzy leaves with a sunflower that's heterozygous for fuzzy leaves. Okay, so fuzzy leaves is dominant, so big F is going to be my letter. I gotta do F again. I'm not good at the F. Okay. True breeding for fuzzy leaves. If it's true breeding, that means that is homozygous because that's all it can give. And it's fuzzy leaves, which is dominant, so big F. Then we're going to cross a heterozygous. Heterozygous means different. So we're going to get a big F. We'll make it real big this time. And our little F with our hook on it, because hetero means different. Okay, so I'm going to have my two big Fs. Two big Fs. Right? And then we're going to have our big F and our little F. And then we're just going to fill it in. So up here, all I have are big Fs, right? Big F, big F, big F, big F. Now down on the bottom, I can have a big F and a little f in this box. I'm getting better at making Fs, I think. And the same thing here, big F and little f. Okay. So if we look at the genotype ratio, I've got two that are big F, okay, these two are big F, right? And these two, see if I can make it a different color. These two are big F, little f. So my genotype ratio, again, is going to be 50-50 or 2 to 2. 50-50 or 2 to 2 or 50%. It depends on the problem as to how they actually will label it. Okay, phenotype. Again, big F is going to give us fuzzy leaves. They've all got a big F, right? This one's got a big F, this one's got a big F, this one's got a big F, and this one's got a big F. So 100% are going to have the fuzzy leaves. So for my phenotype, they all have fuzzy leaves. So it's going to be 100%. So here we can see that now the genotype and phenotype ratio are not the same. And then this last part is looking at if we mated an albino deer with a brown deer, we can actually determine what the brown deer's genotype is. Just looking at him, he could have a genotype that's big B, little b, or if we do it with albino gene, if we say it with A's, it would be big A, little a, or he could be two big A's. But we don't know unless we mate him with a completely recessive individual. This is called a test cross. Then you can see the results here. So here's our albino individual on the side, okay? If the deer is a true breeding brown deer, it only has two dominant genes. all the offspring will be brown. Okay, see how they all have a big A, all brown. If the brown deer, however, has this, it's a heterozygote, it's a carrier. See there, it's carrying the albino gene. Now we have some of the offspring are brown because they have the big A, but some of the offspring are white. They're albinos because they got a little A from both parents. So this is called a test cross. And it's a way that if you need to, for whatever reason, especially with like cat breeds or dog breeds or something, or if you were breeding animals and you need to know that what their exact genotype is for whatever reason, you could do this test cross. Again, I don't know how practical it is, especially with something like deer. Because, I mean, how long is it going to take them to make 4 babies? I don't know. But several years, right? It's not. But if it's a plant or something, it might not take that long. But anyway, this is a test cross. It allows you to determine the genotype of the unknown deer here. We don't know if he's big A, or big A, little a. And again, if we did get albino in the second generation, then we know that he was a carrier. A carrier is someone who has the gene, but they have a dominant gene that masks, you could say, the disorder or the recessive gene.